welcome back to biochemistry class. The second biomolecule that I want to discuss today is protein. For the first part of proteins, I will talk about protein functions, amino acid structures, and classification of amino acid. Okay, you must be familiar with this word protein. There are many functions of proteins. Proteins are a source of energy. Proteins are also sources of various types of hormones. Proteins can maintain our body tissues. Proteins also serve as enzymes. And protein also serve as antibodies. And lastly, protein is a tool of transportation and storage of body molecules. Proteins are big molecules that are vital for organisms. Protein present in all cells. Proteins are polymers composed of 20 standard amino acids. The amino acid chains are linked by specific covalent bond. They call it as peptide bond. The structure and function of proteins are determined by the combination, number, and sequence of amino acids. The physical and chemical properties of proteins are also influenced by the constituent of amino acids. These are food that contains proteins from animal sources like beef, chicken, fish, tuna, milk, cheese, and eggs, while proteins from plants are tofu, nuts, and legumes. If we talk about proteins, we need to discuss first about amino acids. As you know, proteins are big polymers. They consist of amino acids. Amino acids are the building blocks of proteins. Their structure consists of one central carbon atom that are covalently bonded to one amino group, one carboxyl group, one hydrogen atom, and one side chain or R group. The R group or side chains of amino acids are different for each type of amino acid. The differences in R groups determine structure, sizes, electric charges, and solubility in water for each amino acid. There are 20 kinds of amino acids that make up proteins of organisms. They are called standard amino acids. Known to be the 21st amino acid, it's called selenocysteine, and it is rarely found. It's found in several enzymes such as glutathione peroxidase. Amino acid classification are based on R group or side chain. Their common properties are including hydrophobic or hydrophilic, polar or non-polar, presence or absence of ionized group. Okay, let's discuss about the first category. The first one here is non-polar amino acids. These amino acids have an aliphatic R group. They are glycine, alanine, valine, leucine, isoleucine, and proline. They are hydrophobic amino acids. However, proline differs from other amino acids in this category by being cyclic. Anyway, proline has many characteristics in common with this aliphatic group. Non-polar amino acids are commonly found in proteins that interact with lipids. These are the molecular structure of non-polar amino acid. Glycine is the smallest amino acid since its side chain is hydrogen atom. Alanine, valine, leucine, and isoleucine have alkyl in their side chains, while proline has cyclic alkyl in its side chain. The second amino acid group is polar amino acids. These amino acids have uncharged R group. They are serine, threonine, cysteine, methionine, asparagine, and glutamine. They are all polar amino acids. And because they are polar, they are hydrophilic and easily soluble in water. They tend to be found on the outside of the protein. Cysteine is different from other amino acids in this group, since the R group is ionized at high pH at around 8.3, so that it can be oxidized with cysteine to form disulfide bonds. These are the molecular structure of polar amino acids. Methionine and cysteine are polar amino acids that have sulfur atoms. Asparagine and glutamine have amino groups in their side chains, and threonine has hydroxyl group. The next category of amino acids are aromatic amino acids. These are phenylalanine, tyrosine, and tryptophan. These amino acids are relatively nonpolar hydrophobic. Phenylalanine together with valine, leucine, and isoleucine are the most hydrophobic amino acids. Tyrosine has hydroxyl group, while tryptophan has indole ring. Aromatic amino acids are important to determine the structure of the enzymes. Aromatic amino acids are able to absorb UV light at 280 nanometers and is often used to determine protein concentration. The molecular structure of phenylalanine, tyrosine, and tryptophan are like this. As I mentioned previously, tyrosine has hydroxyl group, tryptophan has indole ring. The next category is amino acid with a positively charged side chain or S basis. Lysine, arginine, and histidine are amino acids that belong to this group. They all have an alkyl group on their side chain. Histidine has a nearly neutral charge on the imidazole group compared to lysine amino group. Arginine has guanidino group. Because histidine can be ionized at pH close to physiological pH, histidine often plays a role in enzymatic reaction involving proton exchange. These are molecular structure of lysine, arginine, and histidine. Lysine and arginine have amino groups that can be ionized and have positive charge as NH3+ and NH2+, while histidine has imidazole group and the NH group can be ionized to be NH2+. The last category of amino acids is amino acids with a negatively charged on their side chains or as acids. There are only two amino acids in this category, aspartate and glutamate. They have carboxyl groups on their side chains or negatively charged or acting as acids on pH 7. Asparagine and glutamine are derivatives for aspartate and glutamate. These are the molecular structure of aspartate and glutamate. They both have carboxyl groups in their side chains. You can also see for asparagine and glutamine, they have amino groups or NH2 instead of carboxyl groups. The amino acid names can be abbreviated into 3-letter codes or 1-letter code. The inventor of 1-letter code for amino acid is Margaret Day Hoff. 3-letter code for amino acids are the first 3 letters of amino acids names. For example, alanine is ALA, ALA, lysine is LYS, LYS, histidine is HIS. 1-letter code is somewhat not always the first letter of amino acid names. Like for alanine is A, glycine is G, but lysine is K and tryptophan is W. There are other classifications of amino acids based on their chemical constituents. There are small amino acids like glycine and alanine, branch amino acids like valine, leucine, and isoleucine, hydroxy amino acid or amino acids that have OH groups, they are serine and threonine, sulfur amino acids like cysteine, methionine, aromatic amino acids, phenylalanine, tyrosine, tryptophan, acidic amino acid and their derivatives, aspartate, asparagine, glutamate, and glutamine. And basic amino acids such as lysine, arginine, and histidine. The amino acid is only for proline. There's also essential and non-essential amino acids. Essential amino acids are amino acids that we need in our daily diet. This is because humans are incapable of forming exquisite carbon skeleton. There are 10 essential amino acids. They are arginine, histidine, isoleucine, leucine, valine, lysine, methionine, threonine, phenylalanine, and tryptophan. Then the other 10 amino acids will be non-essential. They are named non-essential since these amino acids are not required in our diet. Non-essential amino acids can be formed by alpha keto acids, by transamination reaction, and their subsequent reaction. The 10 non-essential amino acids are alanine, asparagine, aspartate, glutamate, glutamine, glycine, proline, serine, cysteine, and tyrosine. For the stereochemistry of amino acids, I need to ask you this question first. All amino acids are chiral except glycine. Why? Well, as you know, in the amino acid structure, one central carbon atom covalently bonded to one amino group, one carboxyl group, one hydrogen atom, and one side chain or R group. So, if glycine has one hydrogen atom in its side chain, this makes carbon atom in glycine is not carbon chiral or is not asymmetric. Unlike sugars, amino acids in nature are in the L form. DL nomenclature in amino acids is based on DL nomenclature as in sugar, for example in glyceraldehyde. These are the comparison between DL glyceraldehyde and DL serine. In sugar, if hydrogen groups points left, we call it as L sugar, and if hydroxyl group points right, is D sugar. In amino acids, if amino groups or NH2 or NH3 plus points left is L amino acid, and if amino group points right is D amino acid. The nomenclature of amino acids are somehow confusing. These are examples of amino acids and their nomenclature. So for L-isoleucine, the EUPAC nomenclature is 2S, 3S isoleucine, and for the isoleucine is 2R, 3R isoleucine. The R and S absolute configuration here are similar with those in monosaccharides, EUPAC nomenclature. But you have to remember the names of 20 standard amino acids. The carbon numbering system in amino acids is based on the distance from the central carbon atom. Central carbon atom is an alpha carbon atom, next to it is beta, then gamma, next is delta, and the last one here is epsilon. This is an example as in amino acid lysine. Okay guys, for the next video, I will talk more about properties of amino acids and protein structures. See you on the next video.